everybody, and welcome back to Microbiology Bio229. I am your host, Mary Annarella, coming at you live, well, not live, from during the pandemic. So we're doing a little lecture here on Chapter 6, which is all about metabolism. How cells, bacterial cells, and even your cells make energy from the food we eat. We left off talking about oxidation and reduction reactions. If we use our little Leo goes grr, if you lose an electron, it's an oxidation. So when a molecule loses one or more electrons or hydrogen atoms, we talked all about that last time, it is an oxidation. When a molecule gains one or more electrons or hydrogen atoms, it's a reduction. And so we talked about the overall chemical reaction for aerobic cellular respiration, which is H12O6. I'm going to color code here. Plus 6 O2 yields 6 CO2 molecules plus 6 water molecules plus ATP. You've seen that before. We talked about how it doesn't happen all at once. I need to, again, I'm color coding. And this was to show you that glucose over here is oxidized. It's an oxidation. It loses all those hydrogen atoms, whereas oxygen gets reduced. Alrighty. So we were talking about oxidation reduction reactions. During our Zoom, I mentioned the Khan Academy link, and that Academy link is up there for you. I just want to say a little bit more about oxidation and reduction reactions in regards to cellular respiration right here. Okay, so this part here where glucose is oxidized, right there. These hydrogens here do not go directly to oxygen. That doesn't happen. So forget about that. Instead, they get picked up by a class of molecules called electron carriers. So they get, well, let's say they get pulled out of glucose one at a time by these things called electron carriers. And these electron carriers are these two molecules known as, we need another color. I don't know, what color do I want for them? How about this? NAD plus and FAD. You don't need to know their names. I know you've seen this stuff before, but like I'm not going to ask you, what the NAD plus stands for, nicotinamide adenine dinucleotide. Who cares? No one writes that out. NAD plus, FAD, flavin adenine dinucleotide. Let's just quickly relate this to, here we go, electron carriers. I also call them hydrogen carriers, so let's take a look at why. Oh, and FYI, NAD+, that's one we're talking about, and FA, oh, look at that, FAD, but NAD+, down here, NADP+ down here, that's actually more for, it's not for cellular respiration, but pentose phosphate pathway and also photosynthesis uses NADP+ as an electron carrier. Come back. Here we go. So let's take a look at this. All right, let's look at FAD first down here. FAD is an electron carrier, and it will take on 2 electrons So I'll draw like a little mini arrow going into the main arrow. Oop, that's an E. There we go. And 2 protons. These 2 electrons and 2 protons together make 2 hydrogen atoms, so FAD becomes FADH2. So, oh, and actually, you know what? Let's color code these. I'm liking a little color coding here because I put these hydrogens in red here. FAD will take these hydrogens out of glucose one at a time, or what's left of glucose, in the form of electrons, 2 electrons and 2 protons, and become FADH2. And I'm color coding them red so that you can remember that those hydrogen atoms came from way up here on your left. They're these ones here. It's getting messy there. Let's clean this up. All right. NAD, on the other hand, can bind to 2 electrons, but only 1 proton, like that. And so it becomes, is that the right color that I picked? No, that's not. There we go. NADH. Because we put one of these electrons together with a proton that makes 1 hydrogen atom, and then the remaining electron will neutralize that positive charge there. But again, I'm pointing out that these hydrogens here came from glucose, or what's left of glucose. So, you know, biochemists first described these, and they called them electron carriers because they're looking at them in terms of oxidation and reduction. But really, they're not just carrying electrons, they're carrying hydrogen atoms. And so you're not going to see this anywhere else, but I like to call them electron hydrogen carriers, because it helps us to remember that it's carrying hydrogen atoms that came from glucose. So let's look at this over here in the form of, let's look at FAD here and then compare it to FADH2. Is this process here by this arrow an oxidation or a reduction? Is it an oxidation or a reduction? Leo goes grr. Lose an electron or hydrogen atom, you're oxidized. Gain an electron or hydrogen atom, you are reduced. This NADH and FADH2, this is their reduced form. Their reduced form. And so we call having a lot of these things here, the cell making a stockpile of this, we call this reducing power. Can't spell reducing. Reducing power is having a lot of NADH and FADH2. So for a second here, let's talk about what the heck is going on here. I'll just write all this out again. Gonna love my color coding here. C6H12O6 plus 6O2 yields 6, better 6 there, 6 CO2 plus 6 H2Os. I am gonna go back and color code my hydrogens. So you can see where they're going. What the electron carriers NAD plus and FAD do is that they're hanging out in the cell. FAD, oops, there we go. And they rip these hydrogens out of glucose Or I could do a better job of that. And they become NADH and FADH2. Okay, and then this is called reducing power, having a whole lot of NADH and FADH2. What they do, oh, this is a really crappy looking H there. There we go, that's a little bit better. What they do with these is that they, NADH and FAD, take hydrogens out of glucose and they bring them to the electron transport system. So they bring those hydrogens from glucose to the electron transport system. Now in your cells, that electron transport system is going to be in the mitochondria. In bacteria, it's going to be in the cell membrane. Who cares? So what if they're bringing them to the electron transport system? They bring them to the electron transport system where a crap ton of ATP will be made with those hydrogens. And I'm just writing H's. I'm abbreviating hydrogen with an H. I'm not writing out the word hydrogen. Okay, a crap ton of ATP will be made with those hydrogens in, well, chemiosmosis. That word should be familiar. Chemiosmosis, aka respiration. And we'll go through that process too. When those hydrogens have been used in chemiosmosis, respiration, or also known as respiration, a proton motive force. Those are all parts of that process. When they're done, the hydrogens will bind to oxygen to form water. So let's see, after chemiosmosis, the hydrogens bind to oxygen to make water, and that's way the heck at the end. So this is just kind of an overview. NAD plus and FAD pull those hydrogens out of glucose, bring them to the electron transport system wherever it happens to be in that cell. In bacteria, it's going to be in the cell membrane. In eukaryotes, it's going to be in the mitochondria. And they do their little chemiosmosis thing and make a crap ton of ATP for the cell. And when they're done, those hydrogens join to oxygen to form water. That's the process. But a reminder that having a lot of this stuff is a goal of the cell. The cell likes having a lot of NADH and FAD. The cell likes having reducing power because it means that the cell can do the chemiosmosis thing. So it's really important. So hold this thought for a moment while we go back to our slides. I'm not liking the order that my slides are in here. Every year I try to change them and I try to think of ways that introduce this information to you in the most cohesive and logical way. And then I change my mind and I end up having to mix my slides up. So we're going to go back a couple of slides. Here we go. I want to talk about two different ways to make ATP from ADP. So, one is with substrate level, come back here, substrate level phosphorylation. I need a different color on here. This uses chemical energy released from an exergonic reaction to make ATP from ATP. This is where that coupled reaction thing came in. Those coupled reactions. Hold this. I'm going to go back to my whiteboard. I can't do that. That's right. One of these days I will figure that out, that I can't toggle when I've got a screenshot going. All right, so we'll just write this out instead. I'm not liking this color palette. Let's switch to something else. Here, this is a brighter blue. I like this blue. 2 ways for the cell to make ATP. One way is through what's called substrate level. So at the level of the substrate, phosphorylation. I actually love putting that slide up. I absolutely love putting that slide up when we're in person in classes. In fact, I'm going to show you one more time. I put this slide up, and, and students all see these words. They're like, substrate level what? And they're like, oh my God. Oxidative phosphoryl what? I need to know these things. Yes. So let's break it down. Let's break these words down to get more meaning out of them. So phosphorylation, to phosphorylate something means to put a phosphate group on it. So it just refers to a phosphate group. So adding a phosphate group to ADP, basically to get ATP, is what we're talking about here. So this part here, how to make ATP, it means you need ADP plus its little initial phosphate group going to ATP. So 2 ways that the cell does that are like this. They can do it at the substrate level phosphorylation, and that is when you have some sort of generic chemical reaction. Substance A reacts with substance B to form products C and D. And if this is exergonic and releases energy, we can get ADP becoming ATP. So there's chemical energy that's released in the purple arrow here. So this releases chemical energy. So this reaction releases chemical energy, and the cell uses that to put that initial phosphate group back onto ADP to form ATP. There we go. Substrate level phosphorylation. Think of it as these guys being substrates. That's one way that the cell makes ATP. The other way that the cell makes ATP, boatloads of it, is through not substrate level phosphorylation, but oxidative phosphorylation. I can't spell other words, but I can spell phosphorylation. Here we go. Oxidative phosphorylation. It requires a membrane. And it uses the flow of protons across that membrane to release energy. It uses a proton motive force. So uses the flow of protons across a membrane, or a proton motive force, which we talked about before. I told you this stuff that, what was going to hit the fan and things were going to get intense and busier in this class, in this chapter. Okay, oxidative phosphorylation uses the flow of protons across a membrane, proton motive force, to make ATP that way. Right, so those are 2 ways to make ATP. On the exam, you are going to need to know the difference between these two things. So how does substrate level phosphorylation differ from oxidative phosphorylation? So oxidative phosphorylation, I want to add this too. It's also known as respiration. And for a long time, I was like, is there a difference between oxidative phosphorylation and respiration? Why do we use these two different words? I have looked this up. I've tried to figure out if there is any sort of subtle difference, and there, as far as I can tell, there is not. Respiration is just another way to say oxidative phosphorylation. Oxidative phosphorylation and respiration are the same thing. So cellular respiration, we could call it cellular oxidative phosphorylation. Although I think if we called it that, Bio 107 students would freak out. Like, what? High school students learning this stuff would freak out. Like, we just like the word respiration. Let's use that. As far as I can tell, they mean the same thing. So, okay, so did I miss anything here? Yeah, oxidative phosphorylation uses the energy of a proton motive force to make ATP from ADP. You need a membrane. Substrate level phosphorylation uses energy released from a chemical reaction to power putting ADP and its phosphate group back together. Oxidative phosphorylation uses a proton motive force across a membrane. And you don't need a membrane for substrate level. You do for oxidative phosphorylation. All right, there is such a thing as photophosphorylation, using radiant energy of the sun to create a proton motive force. So obviously this sounds like photosynthetic organisms do that, and they do. They do. And we will study photosynthesis after we finish up cellular respiration. Okay, energy source, let's see, what do I want to, no, I don't want to talk about this yet. So you can put an X through this slide for now. I'm not going to worry about that. Okay, we already talked about oxidation reduction reactions. We talked about electron carriers. Yes, important slide here. So the goals of metabolism, we are revising them a little bit. We had a slide back here, God, somewhere. Where was that slide? Goals of metabolism, harvest energy, make stuff for the cell, and make intermediate precursor metabolites. We are updating that slide. This is revised and revisited as we scoot through all these. Okay, so the goals of metabolism: make ATP from substrate level phosphorylation, make some precursor metabolites, but also generate a bunch of reducing power in the form of NADH and FADH2. So that you can use these for respiration. So one of the goals on the metabolism of metabolism for the cell, any cell, is to generate reducing power so that they can make ATP from NADH and FADH2. So used for oxidative phosphorylation. There we go. So this is revised. Okay. So let's get into the nitty-gritty of this. The main ways that we're talking about aerobic cellular respiration, the breakdown of glucose, is done through some central metabolic pathways, or the CMPs. Might as well take a shot of that. Cells will start the breakdown of glucose through either glycolysis or the pentose phosphate pathway. Cells use only one of these to begin breaking down glucose, C6H12O6. They get broken down partially, and then if cells are going to do respiration, they'll go through what's called an intermediate step, and then they'll do what's called the Krebs, or really the citric acid cycle, tricarboxylic acid cycle. Let's call this the citric acid cycle, or the CAC. Let's take a picture of how, let's look at a graphic of how they fit together. So way up at the tippy top, I'm not going to draw on this just yet, you've got glucose right up here at the top, center tippy top. It will be broken down by either the pentose phosphate pathway on your left or glycolysis. Might as well show you this here. So glucose up here, we need a different color now on this, and it'll be broken down by either the pentose phosphate pathway, although glycolysis is the most common. And I know you all learned about glycolysis before, but in these steps here, there are some enzymes that are required. Like let's say, enzyme Z is required here. If a bacterial cell does not make this particular enzyme, it has to go by route of the pentose phosphate pathway. And if you take a look here, okay, it starts with glucose, C6H12O6. It actually feeds back into glycolysis. Okay, so glycolysis will occur. Delete screenshot. Let's go back over here. Do a different one. All right, glucose will break things down into pyruvates. If an organism is more of a fermenter, those pyruvates will be reduced via fermentation. We'll talk about that later. But otherwise, there is the transition step or the intermediate step, which causes the pyruvates to form acetyl-CoAs. Acetyl-CoAs go into the citric acid cycle here. Whole bunch of reducing power, which is NADH. And FADH2 is used to do respiration, also known as oxidative phosphorylation. Chemiosmosis is a part of that, and you get ATP out of that. So just this overview here. You start with glucose. You do either the pentose phosphate pathway or glycolysis. Most organisms do glycolysis. Glycolysis followed by the transition step, the citric acid cycle, and then respiration. Sometimes fermentation, and we'll talk about that a little later. So this is just an overview. We are going to focus on glycolysis, the transition step, the citric acid cycle, and respiration. Okay, so let's see. Okay, these slides are all out of order. We already talked about this. Respiration uses reducing power from glycolysis, and it's called oxidative phosphorylation. This slide is in the wrong spot. I want to talk about these central metabolic pathways again, and no, I don't want to do that. I want to do this. So we'll be looking at glycolysis, the transition step, intermediate step, citric acid cycle, and respiration. For all of these, you need to know where in the cell, meaning where in a bacterial cell, where in a eukaryotic cell. Let's talk about inputs and outputs for each. Inputs meaning what are the starting molecules. Outputs meaning what are the finishing molecules for each of these. how much ATP in each of these processes do we get out of those, and how much reducing power in each of these, as well as generally what is happening to glucose or what's left of it. In each of these. So all of these. This is intense stuff. Okay, so we'll start off with glycolysis. It occurs in the cytoplasm of the cell. And that goes for eukaryotes or prokaryotes. And it begins with glucose. So that's our input. So let's take a look at the graphic that I have for you here. Here we go, glycolysis. Now first up, I want to show you that at the tippy top here, see those 6 bluish purple balls at the top? That represents glucose, and each of those blue purple balls represents a carbon. So glucose is a 6-carbon molecule. You've already seen what ATP looks like on the left there. It looks like a little Advil pill with 3 phosphate groups on it. Those little yellow balls are phosphate groups. Let's just take a picture of this. That pyruvate way at the bottom there. Gotta get that in there. All right. I'll use blue. All right, so this is C6H12O6 up here. And each of these little balls is a carbon atom. All right, you do not need to know the name of every single enzyme or intermediate. Like, I'm not going to say, what is this molecule here? And you have to be like, oh, glucose 6-phosphate. No, don't need to do that. But check out in this first step, it's a coupled reaction where ATP is becoming ADP. So you know that this one right here is exergonic and releases energy, so that energy must be used because it's coupled here to power this one. So this one must be an endergonic reaction because this one over here is exergonic. And actually, that initial phosphate group goes right here. Another chemical reaction occurs, but check out this third one here. Look, we have ATP becoming ADP again. So this main arrow here must be endergonic, requires the input of energy. And that other phosphate group just goes on the end over here. We have glucose bisphosphate. The little thing looks like a seesaw, doesn't it? With all these carbon atoms here with a little phosphate group on each end. Very cute. Okay, but check this out. We just used 2 ATP. Like, what the heck? I thought we were supposed to be getting ATP out of this. Why did we just use 2 ATP? This seems like a lose-lose for the cell, but it's not. This goes down to the whole idea that you gotta spend money to make money. You gotta buy a lottery ticket for even the possibility that you're gonna win, right? So the cell is spending 2 ATPs here to make more later on. So hold that thought. But we just used up 2 ATPs to make this thing right here. Okay, this next step basically chops this molecule in half. And this one just gets flipped around. So we end up with two of the same molecules, glyceraldehyde 3-phosphate. And in the next step here, some hydrogens get pulled out of there, and we just made some NADH. So we just made two NADHs. That's pretty nice right there. Don't tell me my Apple Pencil battery's low. I thought I charged it overnight. I guess not. Okay, so check out the next step here, though. This is a little different. This chemical reaction here, oh, and by the way, everything over here is the same as everything over here because these two molecules up here are the same. There we go, it's getting a little messy there. But this is ADP here becoming ATP. So this must be releasing energy. This releases energy. So check it out. We just made an ATP here and an ATP over here. So now we're even Steven. We used ATP, we just made two more. A couple more chemical reactions occur. You don't need to worry about these ones. We need to know some generalities here, but check this out again here on this last step. We have ADP over here, and it's becoming ATP, so this chemical reaction right here must be releasing energy, and the cell is using that energy through what? Substrate level phosphorylation. I'm writing SLP there because there's no way I can fit substrate level phosphorylation in this tiny space. SLP, substrate level phosphorylation. This is substrate level phosphorylation. This up here is substrate level phosphorylation. Substrate level phosphorylation. Substrate level phosphorylation. Say that 4 times really fast. So we just made 1, 2, 3, 4 ATP. Check that out. It's very exciting. So we used ATP but made 4 ATP. Net gain, that's what these little dots mean, a net gain of 2 ATP overall. And the end products here are these two 3-carbon molecules called pyruvate. So we get 2 pyruvates out of glycolysis. Delete that. Okay, so inputs. Okay, where's this happening in the cell? Glycolysis happens in the cytoplasm of the cell. Inputs and outputs begins with 1 molecule of glucose, ends with 2 pyruvates, which are both 3-carbon molecules. 2 pyruvates. How much ATP did we get? It used 2 ATP, made 4 ATP for a net gain of 2 ATP. How much reducing power? We got 2 NADHs. And what's happening to glucose, or what's left of it? Well, glucose started off as 1 6-carbon molecule, and now it's been broken down into 2 3-carbon molecules. So it's like we went from 1, 2, 3, 4, 5, 6 into 1, 2 3-carbon molecules that have been oxidized a little bit, meaning they lost a little bit of hydrogen right there. That is glycolysis. Right, so actually, these 2 pyruvate, that looks terrible. People are going to be like, what the heck is that? Excuse me. 2 3-carbon molecules called pyruvate. The next step is called the transition or intermediate step. It begins with the 2 pyruvates. Now, in bacteria, because they're prokaryotes, this is occurring in the cytoplasm for prokaryotic cells. But for eukaryotic cells, the pyruvates get shuttled into the mitochondria. So it's in the mitochondria in eukaryotic cells. Those 2 pyruvates need to be shuttled into the mitochondria. Okay, and remind ourselves that these are 3 carbon molecules. We're just keeping track of the carbons right now. And each one goes through the transition step, intermediate step, whatever you want to call it, which is basically just one complicated chemical reaction. Like this. So I'm going to draw myself a big arrow right there. This is one of those times when a cofactor, coenzyme, in fact, it's called coenzyme A. Co-A stands for coenzyme, A, actually combines with the pyruvate and becomes what we call it an acetyl. No, I'm going to write it a little bit differently. Coenzyme A binds to the pyruvate, and as it does so, it boots out one of the carbons as a CO2 molecule. And it becomes what's called an acetyl CoA. I'm going to color code. Yeah, CoA, not College of the Atlantic. It's coenzyme A. So this acetyl part is actually a 2-carbon molecule because it lost that 1 carbon as carbon dioxide. But as this is happening, NAD, no, if I was using a different color for NAD, NAD plus rips out a hydrogen and becomes NADH. And that's happening in the mitochondria if we're talking eukaryotes. And it's in the cytoplasm of the cell if we're talking prokaryotes. Let's draw this one more time. Maybe I won't color code it this time, but coenzyme A joins with 2 of the carbons from pyruvate, boots out a CO2 molecule, and becomes acetyl CoA. Meanwhile, NAD plus is like, I got to get in on this action here, and I'm going to rip out me a hydrogen atom. So there we go. It's a very busy chemical reaction. That's the transition step. Basically, it's breaking down, it's oxidizing what's left of pyruvate, what's left of glucose, and it's also booting out things as a CO2. Ooh, this is the first time we've seen CO2 used up. Let's go back, way back here. Where the heck is our overall chemical reaction? Ah, there we go. C6H12O6 plus 6O2 yields 6CO2 and 6H2O. Okay, if we use those little tick marks, you know how you go 1, 2, 3, 4, 5, like that kind of thing. We've just made 2 of them, right? So we've accounted for 2 of these 6. Okie doke. So what is next is that the acetyl-CoA is the molecule that enters the citric acid cycle. So let's take a look at that graphic. Okay, glycolysis, talked about that. Yep, happens in the cytoplasm. Oh yeah, we get 6 precursor molecules. I'm not going to ask you which ones they are. Just be aware that there are precursor molecules all along the way here. Okay, pentose phosphate pathway. I'm not going to have you worry about this except when we talked about it back here. Back here. And how it feeds in to glycolysis. And that's for when organisms don't have the enzyme to get glycolysis going. So let's get back up here. See what I mean? I play around with moving these slides around too much. Ah, a word about this. Okay, Krebs cycle is also known as the citric acid cycle. This is what we're just about to begin. So FYI, Krebs cycle is an old name for it. I mean, I guess the person who first discovered and described this part of metabolism, that their last name was Krebs, and if you discover something, you usually get to name it after yourself. But it doesn't tell us anything about it. Like, why don't I just call it the Snooki cycle? That would tell us about as much of what's going on in this cycle as the name Krebs cycle. So this is called the citric acid cycle. Let's take a look at it. And I cut the pyruvate off at the top. But that's okay, because we already went through the transition reaction, and we've got acetyl-CoA right up here. Yeah, let's stick with blue. So this is our acetyl-CoA right here. So the transition reaction just occurred. Coenzyme A gets involved. It boots things out as a CO2 molecule. We did that. All right. There's this 4-carbon molecule named oxaloacetate that combines with the acetyl-CoA, and together they form citrate, which is also known as citric acid. So calling this the citric acid cycle, the CAC, tells us something about what's going on in this metabolic pathway. It makes more sense than Krebs cycle. Like, I don't know what the Krebs cycle is. Why don't we call it the Mary cycle? I like that name better. But the citric acid molecule, citrate, is the first molecule that's made. So it tells us something about this cycle. All right, notice that the coenzyme A gets out of Dodge. It leaves the building. So we're left with citrate. Another chemical reaction occurs. Again, you do not need to know the name of the enzyme that catalyzes any of these. You don't have to memorize them at all. Let's look what's going on here. Now we've got this isocitrate molecule here. And check it out. NAD plus forms NADH, so we get some NADH right there. And ooh, look, one more carbon is booted out as CO2. Over here, next chemical reaction, we lose another CO2 and we make another NADH. So that's awesome. Coenzyme A gets involved for just a moment here, and through substrate level phosphorylation, this is a mistake here. This should say ADP, becomes ATP. So we just made some ATP right there. We've got a 4-carbon molecule left, and check it out. We finally see this FAD guy become FADH2. So we just made a little bit of that. So we're getting some reducing power here. Finally, okay, we got formylate. I'm going to malate, and this last step of the citric acid cycle, NADH is like, ooh, there's another hydrogen left that's available to me. I'm gonna rip that out and become a NADH. And the malate goes back to becoming oxaloacetate, which is available to chemically react with another acetyl-CoA that's hanging out in the cell. So we got some goodies here. We got some goodies out of this whole thing. We got 1, 2, 3 NADH. We got 1 FADH2. We made 1 ATP. I put this on Do Not Disturb. I don't know why I'm still getting notifications. Okay. And we also lost 1, 2 more CO2s. Alrighty. Quickly remind yourself. Let us remind ourselves that, I gotta go back here. Here we go. That when you start with glucose, glycolysis, going through glycolysis here, you get 2 pyruvates, you get 2 acetyl-CoAs. They really should draw 2 citric acid cycles here, shouldn't they? Because there are 2 acetyl-CoAs and each of them go through the citric acid cycle. So buttons. Each one of these acetyl-CoAs is going to go through the citric acid cycle, and each one will give us 3 NADH, 3 NADH from this one over here. Each one will give us an FADH2. Each one will give us an ATP. And each one will spit out 2 CO2 molecules. So we just lost 1, 2, 3, 4 CO2 molecules. Let us go way back up here and add 4 more. So we already had 2 CO2s from the transition step. So we're 4 more. 1, 2, 3, 4. That is 6. So we just accounted for all 6 carbon dioxide molecules. I love it when the math actually works out. Very nice. So all 6 of these carbons that started off as a part of glucose by the end of the citric acid cycle become 6 CO2. In fact, let's go way back. Let's go way on back. This occurs, the oxidation of glucose occurs in glycolysis. And the transition step, I'll call it the trans step, and the citric acid cycle. All that happens first. Now, have you seen anything going on with O2 yet? Nope, because I haven't. Nothing going on there. Not yet. All right, so let's come down here. Okay, glucose is gone now. There's nothing left, but we got some goodies out of all of these processes. And let's write them down. Let's make a little table. Let's put glycolysis in this column, and below it we'll put the transition step, transition or intermediate step, and the citric acid cycle down here. Let's do another column of how much ATP, how much NADH, and how much, what's the other goodie? Oh yeah, FADH2. Okay, remember that these two, NADH and FADH2, they are reducing power. The cell likes those. Glycolysis, how much ATP did we get out of that? Well, I'm thinking back going, okay, we used 2 but made 4, so we got a net of 2 ATP. We got 2 NADH out of glycolysis, and you can go check, you can go fact check me. No FADH2 out of glycolysis. Transition step. Again, you can fact check me. No ATP out of that, but we did get 2 NADH out of it. And before you go, oh, wait, we only got 1 NADH, I said here. But it happens twice because there are 2 pyruvates. So 2 NADH. Oh my gosh, why am I suddenly getting all of these notifications? And no FADH2s out of the transition step. Okay, so for the citric acid cycle, we got 2 ATPs, right? Because we got 1 over here and 1 over here. Because this whole thing happens twice. So 2 ATP, 6 NADH, and 1, 2. Hey. Yep. Things were sticking there. 2 FADH2s. Add them up. 2 ATP from glycolysis, none from the transition step, 2 from the citric acid cycle. We got 4 ATP. 2, 4, 10 NADH out of this whole process, and 2 FADH2s. I will tell you, friends, that the cell just went through all of these cellular gymnastics, all of this stuff that I've been yakking at you for about an hour now, for 4 measly ATPs from substrate level phosphorylation, which I'm not going to write out. I'm going to abbreviate it SLP. That's a pittance. That's like digging 6 ditches and somebody pays you $4. Are you kidding me? No. The cell is not all that excited about this pittance of 4 ATP from breaking down a molecule of glucose, but look at all of this. These are the goodies. The cell's like, oh my God, this was so worth it. I am going to use this to make a crap ton of ATP from oxidative phosphorylation, from cellular respiration. So used to make a crap ton of ATP from respiration in the electron transport. These are the goodies. And remember, I'm going to go back to color coding these. Put those hydrogens in red. Remember where those hydrogens came from. They came from here. They came from glucose. When the cell takes a look at glucose, your cells too, when you eat something, when you eat some carbs, or your cell breaks down complex carbs into individual glucose molecules, your cell isn't like, oh, yum, look at all these carbons, got to get me some of that, yum, those are the goodies. No, 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 no. I mean, it's just going to turn it into CO2, and that's a waste product. The cell goes, oh my God, hydrogens. And there are 12 of them in here. We're going to rip those out by those electron hydrogen carriers, become NADH and FADH2, and we're just going to make a crap ton of ATP out of those in the electron transport system with respiration and chemiosmosis and all that really great stuff. So get ready, because that's what we're going to go through shortly. Okay, so what's up next? Let's do this now. Respiration, aka, oops, not a Q. What am I doing there? Oxidative phosphorylation. It's using reducing power that was generated back up here in glycolysis and the transition step and the citric acid cycle, using this reducing power to make a ton of ATP for the cell. It is done through what's called the electron transport system, or the ETS. I'm never going to write out electron transport system for the rest of this lecture, so just forget that. Okay, oh, and my slide calls it the electron transport chain. I have since changed to the electron transport system because I like ETS better than ETC. It doesn't look like etc. But the electron transport system requires a membrane, so it's the cytoplasmic membrane in prokaryotes because they don't have a mitochondria, but it's the inner membrane of the mitochondria in eukaryotes. As you recall, the mitochondria has, that's not going to work, has an outer membrane and an inner membrane that's all folded. And the electron transport system, it's a series of membrane-embedded proteins that are stuck in the inner membrane of the mitochondria. They'll pass, they are proteins that are in these complexes, and there are complexes 1, 2, 3, and 4. They're made up of protein, many proteins, some of which accept a proton. Some of which accept just an electron, and some of which accept just an electron. Wait, did I just say that? No, I didn't say that right. Some of them, because I'm thinking of the next thing I'm going to do. Why don't I just do it? There, I got rid of the screenshot. All right, I want to show you what the electron transport chain looks like, so we're going to skip ahead a couple of slides here. Here we go. 4 complexes. We've got complex 1, 2. Well, oh, actually cytochrome C is 4, and then ATP synthase is the final one. So actually this one is 4, cytochrome C. These complexes in here, these 4 lumps here, well, not this one, change that. These are made up of many proteins. Many proteins. Some of them accept only an electron, and some accept a whole hydrogen atom. And they'll pass the electrons and protons sequentially from complex 1 to complex 2 to 3. Oh, wait. Oh, there's complex 2. My B. Here we go. I gotta erase all this stuff over here. Cytochrome C is not the third one. It shuttles between 3 and 4. I can't read. Oops. Because I got the sun in my eyes here. Maybe I need to move. Okay. Let's do this again. So we got 4 complexes. 1, 2, 3, and 4. And then ATP synthase, which is right here. Okay, so there are going to be different proteins in these depending on the microorganism. And they basically just transfer electrons from protein to protein until chemiosmosis is done and the oxygen accepts the protons and then the electrons. But there are about 45 more sentences in here that I'm going to use to describe how this whole thing works. So let's go back here and talk about the process. All right, so let's see. I'm going to use orange for the mitochondria. So let's just talk about this going on in a eukaryotic cell. All right, NAD, I don't want to use word for that. NADH and FADH2 will accumulate in the matrix of the mitochondria. Once they accumulate in the mitochondria, they're going to see all these protein complexes of the electron transport system. That's what I'm drawing right now, ETS. All these purple things are electron transport system proteins. They'll accumulate here and say, oh, I brought you all a present. NADH is like, here you go. I'm going to give you this hydrogen atom here. You can take it back. And so it gives it back, and it goes back to being NAD+. No big whoop there. But it gives the hydrogen atom back in the form of those 2 electrons and that hydrogen ion, that proton. Some of these proteins in the electron transport system accept the electrons, but many of them will take the protons and pump them across that inner membrane of the mitochondria. So let's write this down here. NAD, and I can't color code all of this, all these sentences here, but NADH, which got its hydrogen from glucose, gives its hydrogen atom, to the electron transport system or electron transport chain, whatever you want to call it, whichever, in the form of those 2 electrons and a proton. And FADH2 will give it in the form of 2 electrons and 2 protons. But let's just focus on NADH right now. So NADH and FADH2. Excuse me. The ETS proteins, they kind of pass those electrons from protein to protein. I'm going to make a little room here. Like some of them will accept an electron, and then like that cytochrome C and coenzyme Q will pass it from protein to protein, like that. So those ETS proteins will accept the hydrogen atoms from NADH and FADH2, but it splits them back up into their electrons and protons. The electrons get passed from protein to protein, kind of like they're playing hot potato with them. So I got to erase them and get them out of the way so I can draw this other stuff. But the protons get pumped across that inner membrane of the mitochondria. And see, they're in this intermembrane space, like out here. Okay, this is the matrix of the mitochondria in here. But way out here on your upper left, if you check that out, you know, the cytoplasm's out here, because we're talking to eukaryotic cell right now. So, think back, and I'm just going to erase some stuff here so that we have a little bit of room here. So it's not quite so messy. Remember all these NADHs? Back here, we counted up 10 of them, right? So they're going to be, for every 1 molecule of glucose, they're going to be 10 of these, which means they're going to be 10 protons, which means 10 protons are going to get pumped across that inner membrane of the mitochondria. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, and a bunch of these complexes will pump them across. So we have a concentration of 10 whatever per milliliters. We have a higher concentration in here than back in here in the matrix. That difference in concentration is called something. When you have a high concentration of something up here and a low concentration down in here, high concentration in this of protons in this intermembrane space and a low concentration of protons back in here in the matrix of the mitochondria. That, my friends, is called something. It is called a concentration gradient, having a high concentration in one area up here and a low concentration here. And is a concentration gradient stable? No, it's not stable. Nature doesn't favor a concentration gradient. It is not stable to have 10 up here and 0 down here. Nature wants 5 and 5, right? That's how all that that's why diffusion happens. So let's write this down. Okay, this sets up a concentration gradient or more specifically, it's an electrochemical gradient because these are charged particles, these are protons. Electrochemical gradient that isn't stable. So 5 of these up here are going to want to come down in here, right, to equalize the gradient. But there was something I told you way back a couple of lectures ago, that something happens when a gradient that is not stable is equalized and becomes stable. Something very special, very important happens when a gradient is equalized. And that is that not only is it more stable, it's so much more stable that energy is released when these protons equalize the gradient. So let's write this down. Energy is released when a gradient is equalized. Remember, I wrote equalized like equalized like that, and somebody was laughing at me when I did that. Energy is released when a gradient is equalized. That is so freaking important because these protons don't diffuse back like over here. They don't do that. They don't diffuse back over here. They don't do that. They diffuse back into here, into the matrix of the mitochondria in one spot. Clean this up a little bit. And what they do, they technically, this is osmosis because it's across a semi-permeable membrane. They osmose back through one special protein that I drew here to look like a pilgrim hat. This special protein is called ATP synthase. It ends in ase, so you know it's an enzyme. And ATP synthase, ATP synthesize, it catalyzes the reaction that synthesizes ATP. This is the reaction that this enzyme catalyzes. ADP getting its phosphate group back to become ATP. We could write ATP synthase right above, in teeny tiny letters above that arrow. But you know this takes energy. So where the heck does ATP synthase get this energy from? From the protons equalizing the gradient through it. The protons osmose back through ATP synthase. And in this case, well, 5 of them will come back through. 1, 2, 3, 4, 5. As it does that, energy is released, and ATP synthase uses that energy. As they flow through, ATP synthase gets energy to put that phosphate group back onto ADP to make ATP. And what it does is it physically turns this enzyme, it physically turns this protein around. It's a mechanical thing. It is like, have you ever played or seen a kid playing at a pool or the beach and they have one of these little water wheel toys where they pour water or sand over the wheel and it makes the wheel go around? The flow of the protons through ATP synthase is like a wheel. And the protons are like the water or the sand going over the wheel, making it go around, making it turn. This is how hydroelectric power works, right? Like in Holyoke, we got a dam there. We damped that up, And we basically have like a water wheel on the side of the dam so that when we control the flow of the water and let water flow over the dam, it turns the wheel. And that wheel is attached to an axle, right? And the axle on the other end is attached to another gear, and that gear is attached to another gear. And so that's why you have all of these old mills around towns that have streams and rivers, because if you put a water wheel in there, you can make the water wheel go around and attach it to an axle and some other gears inside the mill that'll run your saw. In the case of hydroelectric power in Holyoke, it moves the generator, it moves the magnet so that we can generate electricity. This is the same damn thing. It is the same thing as generating electricity through hydroelectric power. ATP synthase is a mechanical enzyme. Like, you know that this ADP, getting its phosphate group back to form ATP, you know that takes energy. Energy is required to go into that. This energy comes from the flow of these protons up here through ATP synthase. And where did these protons come from? They came back here from NADH. And where did NADH get that hydrogen atom? They got it way, way the heck back here. God, I'm gonna get y'all dizzy here. Back here from glucose, from the food that you eat. Your cells take a look at the carbs, the glucose you eat. They're like, holy crap, look at all of those hydrogens in there. I'm going to rip them out through with NAD plus and FAD are going to rip them out. They're going to bring them way the heck down here to the inner membrane of the mitochondria and release them to the electron transport system proteins. Those protons are going to get pumped across the inner membrane, set up an electrochemical gradient that's not stable, but these babies are going to osmose right back through ATP synthase and physically turn ATP synthase so that it can take, we need a third color here, so that it can take ADP and give it its phosphate group back to form ATP. That happens right here. It's so freaking cool. So let's see, it sets up an electrochemical gradient that isn't stable. Energy is released. I am so proud of this sentence here. Energy is released when the protons equalize the gradient through ATP synthase. Yeah. And then ATP synthase uses that energy to put that initial phosphate group back onto ADP to make ATP, which you know takes energy, and ATP synthase gets the energy from basically this proton motive force. This whole mess up here, this whole process of chemiosmosis, the flow of the protons through ATP synthase and releasing energy, that is a proton motive force. A proton motive force that drives chemiosmosis. And I'm erasing this mess here. Holy crap, it's a mess. What else am I gonna get rid of here? Alrighty. So to reiterate, to come way back here, let's see, C6H12O6, these through the process of glycolysis and the citric acid cycle and the intermediate step, all of these hydrogens get ripped out of glucose by NAD plus and FAD, and they become NADH and FADH2. NADH and FADH2 accumulate in the mitochondria, if we're talking eukaryotes, and they release those hydrogens. They bring them to the electron transport system in the mitochondria, where the hydrogen atoms will split into their electrons and their protons. The protons get pumped across the inner membrane of the mitochondria. That sets up an electrochemical gradient that's not stable. And just like it's like the inner membrane of the mitochondria is a dam holding back all this water, and these protons flowing back through is like water flowing over a dam and turning the wagon wheel, turning the water wheel. Only that water wheel is ATP synthase. That proton motive force, the energy released from that proton motive force, is what gives ATP synthase the energy to put that phosphate group back onto ADP to form ATP, and the cell goes, yay! Now let's take a look at how much ATP that is. Okay, here is the general rule of thumb. Oh, and this whole process here is called respiration or oxidative phosphorylation. ADP gets phosphorylated. It's your new favorite word. Word of the day, phosphorylation. So just how much ATP do you get from some NADH and from one molecule of glucose? Here's the general rule of thumb. For every 1 NADH, you can make, and this is under ideal conditions, it hardly ever happens, but under ideal conditions, you can make a maximum of 3 ATP. So, for every FADH2, and I know this sounds counterintuitive, but FADH2 is actually less efficient at giving up its hydrogen atoms to the electron transport system, you can make a max of 2 ATP. So if we go back here to that table that we made right here, there we go. So we're looking at glycolysis, the transition step, the citric acid cycle. We added up that we got a total of 4 ATP from substrate level phosphorylation, and we got 10 NADH and 2 FADH2s. Under ideal conditions, let me just erase this stuff here. Go. Under ideal conditions, these 10 NADH will make 30 ATP in oxidative phosphorylation, bioxidative phosphorylation, and these 2 FADH will make a maximum of 4 ATP. We add up the 4 from substrate level, 30 plus 4, 38 ATP. Way more than 4, huh? So look, you got 34 out of oxidative phosphorylation. So these are the goodies. Oh, this reducing power makes so much ATP for the cell. But here's the deal. This is for prokaryotes. I do not want to insert an image. It's only 36 for eukaryotes. And that's because it takes 2 ATP to get these pyruvates into the mitochondria. So it's going to be a little less for eukaryotes than for prokaryotes, bacteria. There we go. So that is a lot. Look at this hot mess. It's a beautiful mess. It's a beautiful mess. Okay. By the way, anybody notice that nothing's still happening with oxygen? We look way the heck back up here. Nothing happened with oxygen. I didn't write O2 anywhere. In fact, nothing has happened with oxygen yet. So let's take a look at what is up with that. I'm going to draw the mitochondria, only part of it down here. Let's get back into the mitochondria. Here's a little cross section of it. Okay, so let's see. NADH sees all of these proteins of the electron transport system. And I'm just drawing one set here. Okay, ATP synthase is over here looking like the pilgrim hat. It gives up the hydrogen atom in the form of a couple electrons and a proton. And it goes back to being NAD+. The electrons get passed from protein to protein while the protons get pumped across the inner membrane of the mitochondria. We're going to have 10 of these, so times 10. 3, 4, 5, 6, 7, 8, 9, 10. That sets up an electrochemical gradient that's not stable. So it's not stable to have 10 up here and 0 down here. So 5 of these osmose through ATP synthase, and the cell goes, yay, we can make ATP from ADP now. But let's see, so 5 of these are going to come through, right? 1, 2, 3, 4, 5. So I got to erase 5 up here. Okay, so now I've got 1, 2, 3, 4, 5 up here and 5 down here. Let's neaten these up a little bit. The gradient's been equalized, right? And is it going to go anymore? Nope, it's not. Because, I mean, if this one up here moved through, then we'd have 6 down here and only 4 up here, and that just made the gradient worse. So that's not happening. So crap. ATP synthase is like, hey, come on, y'all, let's do this. Can a few more of you move through so that I can make some more ATP for this cell, because we kind of need it here? And the protons are like, we're kind of stuck. We can't do anything. And the cell's like, wouldn't it be great if we could somehow get rid of all of these hydrogens down here? Then we would have a gradient again. Now we've got 5 up here and 0 down here in the matrix of the mitochondria. And the cell's got a really neat trick to do that. The cell's not like, oh, hey, let's just shuttle all of these protons out of here. Nope, that doesn't happen. That's way too much work. Instead, the cell's like, we got this. If we could somehow bind these protons to another molecule, they wouldn't be here anymore. So the cell's like, all right, yep, I got this. The other molecule is like, I offer myself as tribute. And that other molecule is O2. That has, just been has just diffused right into your cells, diffused into the mitochondria. What happens is that oxygen is like, hey, I'll bind to these protons, but we got to have some electrons to go along with you. And meanwhile, the couple of electrons that are up here are like, where the heck have you been? Right? So the protons and electrons will get back together, bind to oxygen, and become, wait for it, H2O. And let's see, we got to balance this equation. Let's balance this. Let's balance this. We got to have 4 of these and 4 of these. Let me get rid of this circle there. There we go. Yeah, does that work? No, I got to change this a little bit. To make 4 H2O. And actually these should be red, right? And that should be blue. I'm going to get this color coding metaphor correct. No, that should be a 2. 2, sorry. And that should be red. There we go. Now it's a balanced equation. Okay, so if we used up, there were 5 of these here. 1, 2, 3, 4, 5, blah, 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 blah. Let's leave 1. Now we have 5 up here, but only a concentration of 1 down here, right? So that means 3 more of these can move through. Now it's like, now we got rid of 4 of these protons. Now 4 more can flow through. So what oxygen does is that it keeps the gradient moving. It keeps it flowing by essentially removing those protons and electrons from the matrix of the mitochondria and turns them into water. It's like magic. So we call oxygen. Oxygen is, it happens way at the end. We call oxygen, and actually, if you look up oxygen, molecular oxygen on its wiki page, and yes, it has a wiki page, it will say oxygen is the final electron acceptor in oxidative phosphorylation. Phosphorylation. It keeps the gradient going. Because if it's not there, if it's not there to bind to those protons and electrons after chemiosmosis, then we'd get 5 coming down. We'd have, the gradient would equalize, and we'd have 5 protons down here and 5 up here, concentration-wise. And then no more protons could flow. And if there are more NADHs waiting around saying, hey, come on guys, here you go. Here y'all, let me give you those hydrogen atoms. They're like, nope, can't do it because it gums up the works. No more water will flow. The dam is full and no more water can come down and there's nowhere for things to flood. And this is where that metaphor is breaking down, isn't it? Right? So if oxygen isn't around, the cell can't make more ATP, and NADH and FADH2 can't give up any more hydrogen atoms to the electron transport system. So a cell can't make any more ATP that way. It's screwed. So if no O2 is present, and you're an obligate aerobe like we humans are, NADH and FADH2 can't give any more hydrogen atoms to the ETS. No more ATP can be made. And that makes a cell sad. So meanwhile, but the cell's going, well, crap, we need more ATP. Isn't there something we can do, NADH? NADH, do something. They will do something else instead. And this is like a last-ditch effort for your cells and for obligate aerobe cells. Instead, NADH will donate its hydrogen atom to a different molecule. It can't give it to the ETS anymore but it will donate its hydrogen atom to an organic molecule. So instead, NADH and FADH2 will give its hydrogen atoms to an organic molecule. To an organic molecule. This is called fermentation. It's when an organic molecule is the final electron acceptor. Usually, it's pyruvate. And the deal that I know you all learned about fermentation with in earlier biology classes is that you get a tiny bit of ATP, but you don't go getting those extra 34 ATPs. You only get like 2 to 3 ATP. So, but only get 3 to 4 ATP. So it's not much, but it's a last-ditch effort for a lot of cells. So, best bedtime story ever. Oxidative phosphorylation. It's what's for dinner. It's how your cells make energy. It is how E. coli cells make energy. It is how obligate aerobes make energy. But it hinges upon having O2 present to bind to a bunch of these protons and their electrons, which are over here being played hot potato with. It hinges upon O2 being present as the final electron acceptor. This is the process of respiration. Respiration involves this proton motive force, which is the flow of those protons through ATP synthase to equalize a gradient. And together they form water. No O2, no O2, you don't get any ATP. You don't get any ATP, a cell's going to die. A lot of cells have a backup plan, and they can get by with fermentation. Multicellular organisms like humans cannot get by with fermenting. All right, we can't do that. Single-celled organisms, no problem. E. coli is like, oh, I can't do aerobic respiration. That's okay, I can ferment. And, 2 to 3 ATP molecules is enough for a single-celled organism to get by. They're okay, but it's not enough for your cells. I should point out that fermentation by bacteria result in a lot of different acids. And I talked about that, oh gosh, a month ago when I was talking about how people will use a bacterial rennet to make cheese, and the different acids that are made as a result of the fermentation process by these different bacterial cells will flavor the cheese, that sort of thing. So you get a lot of different acids. If it's by yeast, and yeast loves to ferment, it results in ethanol, right? But human cells, by human cells, I think this is true for all mammalian cells. By humans, we produce lactic acid. We are not ethanol producers when we ferment. We get lactic acid, and that is what causes muscle cramps. If you've ever had a muscle cramp, it hurts like a you-know-what. Muscle cramps result from muscle cells not getting all the ATP that they need, such as when you are running in a marathon. If your heart if you haven't trained to the level where your heart can effectively pump the oxygen in your blood to all of your cells, all of your muscle cells, your cells will start fermenting. They'll produce a little bit of lactic acid, and muscles are protein, people. So when you get a bunch of acid building up on your muscle proteins, those muscle proteins will denature a little bit and start to lose their shape, and they get as hard as a rock, and you can't run anymore. You can't run until you can rid those cells of your of lactic acid and get them some more ATP. And they say the way to do that is, well, for one thing, train to the level at which you want to perform. Don't go running a marathon when you've never run more than, a 5K in your life. And also stay very hydrated, because remaining hydrated allows the lactic acid to clear a little faster. So yeah, muscle cramps caused by the fermentation process in your cells. But in bacteria, we tend to get a lot of different acids. Shoot, I think I finally ran out of battery with my pencil here. I did plug it in for a little while. That's why I did this lecture in 2 parts. But this is a lot. I know it's a lot. So please make sure you take really good notes, and I can take you through this whole thing again, there's a little Zoom session. So be aware of that. But let's just go back and look here, see if I missed anything. Okay, we added all this up. Yes, we talked about how much ATP you get in respiration, oxidative phosphorylation. Talked about this. Yeah. Here we go. Total of 38. Talked about the electron transport chain. You got to know what ATP synthase is. You need to know this whole, you need to be able to describe this whole process of oxidative phosphorylation, chemiosmosis, respiration, whatever you want to call it. You do not need to know the names of all of the different electron transport chain proteins. I'm just putting up this flavoproteins, flavoproteins, quinones, cytochrome C. These are words that are going to be a little familiar to you. Like, I know a lot of people take quinones and like coenzyme Q. You can get that at Whole Foods, and a lot of people feel that it's like this mitochondrial cocktail for you, and helps people with certain metabolic issues. But you don't need to know the names of all of these. I just wanted you to see them. Yep, the electron gets transferred until oxygen accepts a hydrogen atom, becomes water. I showed you this sweet little graphic. We talked about fermentation. Organic molecule is the final electron acceptor. You do not get as much ATP as in respiration, but you do get some interesting end products like lactic acid if you're human, ethanol if you're yeast. Yeah. I wanted to talk about this. This, you're going to put a giant X through. I'm not able to do that this year with a remote, mostly online course, so I cannot do this. So take a little picture of this and see if my pencil has enough juice for me to go ixnay that. So I tried to find this slide before I even uploaded it to Moodle, and I couldn't find it in there, and lo and behold, here it is now. So I'm saying, sorry, I can't drop one of your grades this semester. So keep up with all this stuff. We will answer questions about this later. Nice to see that my pencil's working again. All right, photosynthesis will be for next time. But this is a lot of stuff for this exam. Everything that we did from last time, the middle of chapter 4, chapter 5, and chapter 6 will be on the upcoming exam. So I will see you in our next Zoom session, and I hope you have had a lovely long weekend, and I'll see you next time. Bye-bye. Have a good day.